leads back again to the view which scientists have, which they say, well, it's not our job to solve all society's problems. It's some other department, some other ministries, and so on and so forth. But that begs the question, because the principal point is, no matter how complex our environment and society is, science and technology poses very interesting solutions to complex problems in a variety of ways. And I won't go through this point any further, but just to quote what Venki Ramakrishnan said some years ago, when asked what kind of science should Indian scientists pursue, he characteristically denied that he was an Indian. He is an American citizen in the UK, holding a UK citizenship, and therefore said that he cannot tell others what to do, and then characteristically went on to say what others should do, which is, he said that you should do one of two things, which is to either work on world-class problems and be of world-class quality, or take on problems from your surroundings, which everyone in the world regards as world-class, and partners with you to come and work here, just as you partner with people across the world to work on currently defined world-class problems. There, I think there's an incredible opportunity in India, not just in a version which you say that interesting problems are applied problems, but both basic and applied problems in our environment are enormous. Again, I won't go into what those are because of the time we have, but also particularly in the life sciences, those problems transcend a divide much more easily between basic and applications, and they're very, very interesting, right from molecular biology at its finest level of granularity to systems biology. There are fantastically exciting problems in our ecosystem, which can be very valuable. So that's what partnership is about, and that's what we need to do. In that context, where does EMBL come? EMBL, I mean, as you can see from the small set of people over here, and those of you who have gone to Heidelberg and met people elsewhere in Europe and all over the world from EMBL, it's just absolutely stunning what EMBL has done to science all over the world, and to Europe in particular. It's a powerhouse, of course, of science in itself, but its impact has been transformative in all over Europe. And its vision of looking at science as a global enterprise is what brought them and us into discussions. Where can we go ahead in this? Now, there are formal models of various kinds, and I'd like to put on the table for your views and discussions between now and a few months from now about how those partnerships should go forward. Basically, structurally, there is an option for us to formally partner with EMBL in a manner where their core facilities in Heidelberg and Cambridge, in Grenoble or Hamburg? Grenoble. And Monte Rotondo and elsewhere, and other partner labs which they have, all of those can be in some ways available, in all ways available to Indian scientists to partner and use in many ways. And that completely transforms the kinds of scientific questions you can address, unlimited by the technology and the resources available and the facilities available. A second component potentially of such partnerships, which EMBL has had with many other countries, is something for us to discuss and think about, is having partner labs. And they've done a phenomenal work in that direction in multiple countries. One example is in the Nordic countries, where a competition resulted in multiple laboratories in the Nordic countries being partner labs, and that's something of a model which we could have, by which laboratories in India are more formally connected with EMBL, so that there's a seamless movement of EMBL scientists to these laboratories and back, but also there is a certain bar in terms of the expectation of science and the management and the kind of goals these laboratories can take, not only as excellent centers, but as hubs which reach out to a broader local environment and cause their have a big impact there and a transformation there. So these are the kinds of models available, and that's something which we should in the breakout sessions talk about and discuss and get your views on how it can be done, how it what are the pitfalls, what are the advantages, and our colleagues from EMBL and the rest of us from all over India are here today and tomorrow to discuss that, so that'll be really valuable. I won't say anything more for now. There's lots more to discuss, and I'm already about 7 minutes ahead of time. And I'll now hand over to Ian, and he will give an overview of EMBL's current role and directions, and then we'll have a presentation by Reiner on EMBL's core facilities. Ian, thank you.
So thank you very much, Vijay. Thank you for the introduction. I think there are many common points in what we're going to say. I will probably try to say things in a slightly different way from the EMBL point of view. But first, I'd like to welcome everyone here. I know that many of you have traveled from many different parts of India. I realize that there are several old friends here, but there are also many people who I haven't had the opportunity or the pleasure of meeting. And so please do take the opportunity over the next couple of days to introduce yourself, to tell myself and my colleagues what you're doing, etc. And my colleagues are here not only to give their talks, but to talk to you and to potentially, and this is one of the hopes of such meetings, to develop the possibility of collaborations between scientists or even institutions. So I'll start by telling you a little bit about EMBL. So EMBL is an intergovernmental research organization. We operate from 6 different sites. So we have 6 different locations. So we are already, if you like, a network, although we're a single laboratory. We have in total about between 1,600 and 1,700 staff. The laboratory is very international. There are people from 80 different nationalities currently working within EMBL. We are the only European level, in fact, in the world, we're the only intergovernmental research laboratory organization. And we not only engage in research, and obviously our reputation is often based on the fact that EMBL is a really internationally leading research laboratory, but we are also based on strong interactions with the communities in our member states, which come about not just, they do come about through research collaboration, but they go beyond that because we provide research infrastructures, we develop training programs, we have intensive training programs at EMBL for external people. We have facilities which external users use as researchers, and we also interact a lot with our member state governments through their representatives, like Vijay in the case of India, although India very soon will be a member. We're not there yet, and there are a few administrative hurdles over which we will undoubtedly fall and then get up again and try again before this happens. But we hope in a relatively short time India will be an associate member state of EMBL. Currently, as Vijay said, we have 23 European member states, and we have 2 associate members. Australia was the first, and Argentina the second. These associate memberships have been very successful, and it's those models, the models by which we interact with these different countries, that we have discussed in detail in discussing India's associate membership. internationalization is completely part of science. And there is a quote from John Kendrew. The European intergovernmental research organizations were set up in the post-war period. And so one of the goals of Europe's governments after the Second World War was to avoid future wars. And so one of the things they did was to set up ways to collaborate. And John Kendrew's quote was that science has always been the most developed international activity that there is. It's as simple as that. That's why science was one of the areas in which Europe decided to get together after the Second World War. And I think it also gives us the opportunity globally to tackle societal challenges, as Vijay has already mentioned, and I will go into this in more detail. So India as a potential associate member state is of course of tremendous interest to EMBL as an organization, but also as Shashi and Vijay could see when they discussed membership with the member states, with all of our member states. India its contribution to global science and technology is already impressive, but it's obviously steadily growing. And it's growing on the basis of a young and very lively population. The average age in India, I believe, is currently 28. The average age of Germany, the country in which our headquarters laboratory is located, is 47. That makes a difference to how quickly you can do things, how enthusiastically people engage in different activities. And EMBL was set up, and this again is a quote from John Kendrew, EMBL is a place for the young. It was set up, our faculty is largely based on hiring people after postdoctoral work and giving them a fixed term position for 9 years, after which they will move on to work usually in one of our member states. Most of our group leaders and team leaders moved on back to the member states. And so we are a place which is used to developing and to mentoring young people to perform at the very highest standard. And EMBL is and has been for 25 years measured as one of the 10 top research institutes in the world. And so one of the secrets is to set up structures which enable young people to start and to succeed very quickly. And I think this is one of the attractions which you ought to take advantage of. And this may be taken advantage of through, for example, the Partnership Lab program. But there are many other aspects to it I'd like to discuss in which we will collaborate. Now, we discussed over several years which areas we should begin with, which areas we should focus on as the beginning of our collaboration. We wanted areas that were important to both sides, that had significant expertise on both sides, and that would significantly benefit from us working together. So the three initial areas that we chose were bioinformatics, structural biology, and imaging. Is this not working? Into my nose. Okay, good. Very good. The problem of having a big nose is the microphone never gets close to your mouth. Anyway, so essentially these are three areas where we really see that that science and technology is moving extremely quickly. There are three areas where we are strong not only in research but in infrastructure development, technology development, and in training. And training is obviously going to be a large component of our interaction with India, because everything that we do here should have a multiplier effect. It should not be based on helping a few people, it should be based on helping as many people as possible. possible. So these areas, obviously, bioinformatics, I think, is obvious to everyone. In the era of genomics, and particularly human genomics, the potential for impacting human health is enormous. But the potential is not limited to human health. I'm not sure if Kath or Rolf will discuss this later, but among the heavy users of the data resources of EMBL EBI are people from very different areas, including agriculture, livestock breeding, food science, areas which are critical to develop globally in order to feed our growing populations. Structural biology is a tremendous way to gain mechanistic insight into how organisms work, how organisms interact. And again, this is used in many applied ways. And again, I don't know exactly what Stephen is going to talk about, but one of the things that Stephen has been able to develop in recent years through his basic work on influenza are pipelines towards new types of drugs to fight influenza. And structural biology gives you the kinds of insight which are very useful to move towards drug development and drug designs. And thirdly, imaging. Imaging, and Reiner and Lars Hufnagel will talk about imaging at the light microscopy level. Kirsten will talk about and Stephen will talk about imaging at the structural level. These different areas and their integration across scales is, I think, the way of the future. To look to see things happening in real time in living organisms, including looking to see things going wrong in human beings who are suffering from disease, whether it's genetic or acute, is going to be tremendously useful to not only scientists, but all of society in the future. So looking at these three areas, I think we have chosen areas where we will be able to work together, and where I look forward to success in having multiplier effects on both sides from our interaction. So I just conclude by reiterating my enthusiasm and my colleagues' enthusiasm for working together with you. We have a lot of expectations from this collaboration. I would like to specifically thank Vijay. He has been a pillar over several years of discussion. He has moved this project forward. His commitment to internationalization and to science is absolutely wonderful. I wish all of our member states had people in charge of government departments who were as equipped to do an excellent job as is Vijay. So I thank him. I also thank him for and I hope that we can use the next couple of days to get to know one another better, to look towards the development of new collaborations, and to look towards discussions of ideas and possible ways in which we can work together in the coming years. Thank you very much. Okay, so the next speaker is Reiner Peppercock. Reiner has a dual role. In fact, he has as do many of my senior colleagues, he has multiple roles at EMBL. He is in charge of a group of core facilities, user facilities in multiple areas, with the exception essentially of structural biology and bioinformatics, which have much larger infrastructures. He also is an extremely good cell biologist, and I'm sure many of you know this. But he is a developer also, and a very advanced user of imaging technologies. And he's going to talk today, I believe, about imaging technologies and the core facilities at EMBL.
Good morning, everybody. It's a real pleasure to present the core facilities that we have at EMBL in Heidelberg. As you have heard from me, and we have more services at EMBL, and I think my colleagues will, in the next 2 days, explain much better what we have in other places. I'm extremely pleased to hear from okay, better? I'm extremely pleased to hear from Vijay that this course that we organized many years ago had such an impact that it actually led to something like this international course in Bangalore. And I must say, I was really at the time already quite extraordinarily impressed on the quality of science and also on the interaction we had at that time. And I hope we can actually continue along these lines with core facilities or with other scientists in the future through this collaboration. So let me focus on the core facilities that we have available at EMBL in Heidelberg. What I will show you is give you a brief introduction, how we organize them, what they are doing, how you can access them. And towards the end of my presentation, I will show you a brief video where we have selected 2 core facilities, the light microscopy facility and the electron microscopy facility, where you're getting a short tour on what we have available there, and you also get to know some of the people who run it. Initially, we had thought to do actually a live transmission, but we got a bit scared that eventually the connection would fail. So what my colleagues have done, they have actually mimicked a live transmission. So what you will see is a not very perfect. I mean, and it's quite good, but it's not a perfect and not too much cut tour through the core facilities. So there may be perspectively a few things missing. So the core facilities we have available in Heidelberg, they are presently 8 of them. And the light microscopy core facility is one of the oldest one, where we provide service in light microscopy, electron microscopy, what reveals the name. It's predominantly in cellular electron microscopy, but also a lot of correlative methods. We have a service in flow cytometry, and our youngest core facility is the metabolomics core facility, where we are providing a service on a mass spec based analysis on either lipids or on small metabolites. Our genomics core facilities has a strong focus on the next generation sequencing methods and bioinformatics, and related technologies like single cell sequencing and analysis. The proteomics core facility, based on mass spectrometry, again is doing any kind of protein on a large scale and also on more detailed scale protein analysis. And we have also a protein expression and purification core facility, where we are producing proteins, sometimes very challenging proteins. It's very important for our structural biologists. And we also have a service for biophysics, where these proteins are analyzed. And a kind of, I would say, in more exceptional for many institutes, core facility is our chemical biology core facility, where we have the possibility to screen small compounds in the functional assays, very often of them of cell-based assays, in order to identify compounds that have the potential to be developed eventually in the future into a drug. And for this, we also have offering a service in order to further develop these compounds by chemical methods. So these are the heads of the core facilities. So you can see when the years when they started. Some of our core facilities are already quite long established. And we also have a turnover in core facilities. So the people here labeled with the stars, that we have more recently recruited them. Some of them, they are already the third recruits to these respective core facilities. Some of the core facilities heads have also joint appointments with research the units, and like myself with cell biology, cell biophysics, and Mikhail Savitsky with the genome biology, Yannick with the cell biology, cell biophysics, and Theodore with the structural and computational biology. This enables us in the core facilities also to introduce latest technologies as they are developed in these respective laboratories. So what are our missions? Of course, to provide a high quality service, and we offer access and expertise to cutting-edge technologies. But what is very important is it is always based on EMBL scientists' objectives and needs. So this can change over time because we have a turnover system and our scientists are changing. And when you see a core facility nowadays, it may be in 4 or 5 years, there may be additional technologies coming up, and some technologies may eventually be ceased. We also contribute to internal and external training courses and workshops, and I will give you some examples of that. And very importantly, we contribute to technology developments in collaboration with industrial partners, and in order to stay at the cutting edge in our technology. And we develop and participate in community building. So just an example how we interact with the industry. Typically, we have users that have a certain problem, we have certain expertise in the facility, and together with our partners, we are then configuring, for example, equipment or designing a certain method, which is then set up and tested in the core facility. And once this is completed, we are offering a service to our users, which allows then us to give feedback to the industrial partners for further improvement of the technologies, and also feedback to the facility in order to improve and gain some new expertise. And we think with these modalities, we have found a way to stay at the cutting edge, at least at the level where commercially equipment is supporting our services that we are providing in the core facilities. So what are our service models? We have one which is it's more like access, like the light microscopy, electron and flow cytometry, where we teach and use the users, we consult them, and of course we are taking care of the equipment. And very importantly, it's the users that are carrying out their experiments themselves. And we have then more service-based core facilities, yes, okay, where the facility staff carries out the experiments. Again. And of course, to a certain extent, we also offer the people to come. Sorry? Yeah. Okay, yeah, it's okay now. Okay, I know I have to, maybe I do it like this. So we also provide a service in if desired, in teaching the methodology, consultation, and also the equipment. Next slide. So our core facilities are not only in use by internal users, but also by external users. And you can see here with the green bars, what is the fraction of external users. We are open to a certain extent when we have capacity, and on the average, it is about 20% of our users are external users. Of course, there is a variation in different core facilities depending on the service that they are providing. Excellent. Most of them come from our member states, and there's only very few that come from non-member states or across the world. So how can you access the core facilities if you want to use them? I mean, this would, for example, in the future be something for Indian scientists, how to use it. Typically, the interested people explore which services are offered. Then they contact their respective core facility head, and in order to discuss the availability and the capacity. And very important, they also identify very often possible scientific hosts. But this is something that is not only a collaboration between core in the level of the core facility, but also this very often triggers also scientific collaborations. Then we develop a project plan, including the detailed scheduling of the visit. Then you can visit the facility and conduct the experiments, also data analysis. And even when the scientists have gone, they can continue data analysis after their visit as e-visitors. And very often they have access to specific software, for example, via virtual machines. And can you go back one more time? I should also say we have also launched a program which we call Christian Boulin Fellowship Program in memory of Christian Boulin, who was the head of and the founder of the scientific core facilities, and where we give support for travel and accommodation when people are visiting core facilities or services of EMBL. So we also make a contribution to training, and on average we organize 15 practical courses, also at the international level. And here I've just shown a number of examples. This is not exhaustive, that of the various core facilities. Typically, we have some collaborative institutes or some collaborators and also some sponsors. They can be either EMBO, a very prominent one, or it could also be our industrial partners. So yeah, go ahead. So we also have some facility networking, like for example, we are members of the European Cytometry Network, protein production purification, yes. And we have been founding the European Light Microscopy Initiative, which is now since more than 15 years, is organizing some exchange of information, mostly via annual meetings between industry, users of core facilities and core facility heads. And we are also involved in initiatives like Euro Bioimaging and Global Bioimaging. So we have also a more formal collaboration with a number of core facilities of our member states, including, for example, the Institut Pasteur or Institut Curie in Paris, or the CRG in Barcelona, also the CSF in Vienna from Austria, where we are discussing the best practice and management in core facilities, and where we also exchange modalities of training. We do a lot of benchmarking in order to see how good we are, and we also do some technology scouting, something that is very important in order to stay at the cutting edge. So something that we are currently developing for the future is very often cross-facility correlative workflows. It is that a certain core facility like the light microscopy core facility, of course we have our strengths in light microscopy, but very often now our users are demanding an analysis that goes beyond, for example, like single cells phenotyping, followed then by single cells transcriptome sequencing and so on. So we are developing in collaborations with our core facilities some interactive and interactions of workflows. Slide. This requires, of course, a lot of data management, analysis, integration. Can you go ahead? Okay. And where we are integrating our light microscopy data together with the flow cytometry, proteomics, metabolomics, and genomics and transcriptomics data, is in order to extract the information that is required by our users. Good. Now we can leave this and load up the video. So after this introduction, I would like to show you now the little tour through the light microscopy and the electron microscopy core facility.